work I'm going to present today is work that was done by a very talented postdoc, Dr. Kevin Liu, who's now a PI in China and was unable to present today. But he did much of this work with me at McGill, and it's also an extension of his PhD work with Corey Lal and Piotr Kozlowski at UBC. So many of the PIs in their talks have given overviews of the center. So I just devoted one slide to describing what we have at the BIC. Our scanner is a 7T PharmaScan single channel 1H system. It's 17 years old, so it's a relatively old system, but it's been a workhorse for us for many years. And it's widely used by some preclinical investigators at our site. And it's located at the McGill Genome Sciences Center. So it's an adjacent to the bioinformatics core. It's not at the Montreal Neurological Institute. And I have some other example images from research we've done with other PIs at the end of the talk, if there's time for that. So Vlad has already described myelin water imaging. Perhaps I won't go into too much detail about that. He described that myelin water imaging has often been done using multi-echo spin echo or GRACE or gradient echo based, spoiled gradient echo based methods where multiple echoes are collected after an RF pulse, and the exponential decay can be decomposed into multi-exponential T2 or T2 star decay to extract myelin water fraction as the ratio of the fast decaying, in our case, T2 star component to the ratio of the myelin water plus the intra-extracellular water. And as it pertains to this talk, there are a variety of techniques that can be used for extracting myelin water fraction. Vlad described RPCA, which is a non-model based approach. For multi-echo T2 star data, non-negative least squares and non-linear least squares have been applied. More recently, Lee et al. In CAMMRI in 2021 applied an artificial neural network, which was quite interesting. And our work will focus on some of Kevin's work, which was also done at UBC on a method called same echoes and the self-labeled encoder-decoder, which is the subject of this talk. So broadly, the fitting of the myelin water fraction decay signal can be broken down into two approaches that are generally seen in the literature. There's model-based approaches. Two of these that are popular for T2 star myelin water fraction mapping are non-linear least squares, shown here with a fairly parsimonious three-pool model, where the A's in this equation denote the amplitude of each pool, and the T2 stars that denote the relaxation properties of the myelin water, the axonal water, and the extracellular water. So this is a simple and easy to implement decay model. You can implement it with some trust region reflective fitting at a voxel level, but it's susceptible to noise and it's susceptible to initialization of the T2 star and the A parameters in your model. More sophisticated methods have been developed using artificial neural networks that essentially build up a synthetic dictionary of decays. In this case, 1 million decays for 24 echo data in our case. And although the topic of my talk is not artificial neural networks, we were asked, this is based on a paper in Neuroimage, we were asked to do this. So I'll show some data from the artificial neural network approach too as a comparator. This essentially trains based on a large number of simulated synthetic decay profiles. And then the trained model can be applied to unseen experimental data to extract the myelin water fraction, the myelin water T2, and the intra and extracellular pool sizes and T2s. Kevin was particularly interested in doing this in a non-model based approach, so model-free data analysis. And the reason for that is that he said, well, tissue microstructure is variable, assuming that parsimonious three-pool model may not be appropriate. So let's try to do this in a model-free way, in an unsupervised way that doesn't necessarily require training data. So the traditional way to do this, which was introduced, as Vlad said, by Whitall and McKay, 1989, is to use non-negative least squares with, for instance, 40 logarithmically spaced T2 star exponential functions. And Kevin came up with, during his PhD at UBC, sort of an augmentation of the NNLS approach, which is fast and which produces improved results in terms of fitting accuracy and lower mean squared error relative to a gold standard data set. And what he basically did is he said, one of the problems with non-negative least squares is that your SNR may not be high enough to resolve the myelin water in the intra and extracellular water pools. So can we use information theory to propose something called a resolution limit for the separation between, for instance, the fast decaying myelin water spectra and the intra and extracellular spectra? And through his method, same echoes, he showed that this could improve based on traditional NNLS. So the work he did in my lab with a mouse model that Vlad described used this architecture, basically a self-labeled encoder-decoder, which consisted of four neural networks, one neural network for each of the T2 star decay parameters. The T2 star and amplitude parameters are encoded into a latent space through a dimensionality reduction approach. So basically, the 24 echo decay signal and the magnitude data is an input to the encoder layer, which has, for those who are really interested, it's a 256 by 120, 24 by 256 by 128 three-layer neural network, and then the decoder layer is actually physics-based. So this model wasn't completely, this fitting algorithm wasn't completely model-free, because in the decoder layer, we extract the individual pool sizes in myelin water for each voxel, and this is done iteratively, and it's iterated until the mean square error is minimized. So I mentioned although this algorithm was quite powerful, it could fit T2 star-based myelin water fraction mapping with our 100 micron data in about 2.5 minutes. It still required this decoder layer, and Kevin was worried about this because this requires initialization of the parameters for the fit. So he resorted to taking the work he did during his PhD at UBC and applying it to T2 star-based myelin water fraction. And this slide's a little bit busy, but essentially what his method does is it says, if our imaging acquisition with multiple echoes has a given SNR at each echo and a given number of echoes, how many distinct pools can we resolve? And once we know the number of distinct pools we can resolve, for instance, the value here was approximately 3 based on the SNR in our multi-echo gradient echo data. What's the separation in the pools in the spectral domain that we can resolve? So if this is smaller, that's better, because we can resolve individual pools that are more tightly grouped together. And Vlad's data, for instance, was fixed brain tissue. So there's this phenomenon in the fixed brain tissue where what we saw was that the T2 star relaxation of the myelin water was a little bit closer to the intra and extracellular water in some cases. So this is just a diagram of the resolution limit formalism. Basically, what we're trying to do is a balance between the stability but oversimplified 3-pool model and the comprehensive but unstable traditional NNLS. And I kind of described this information theory formalism, so I'm not going to really belabor that. But I think this slide, which Kevin produced, is kind of helpful to understand what happens. You use information theory to pick the resolution limit, to pick the number of resolvable peaks and the corresponding resolution limit. That is the spacing between the T2 star peaks and the spectral domain. These peaks are then embedded into a basis spectrum to yield training data, and this was used to train a neural network. Now, for our data, Kevin didn't do all of this. He simply used the information theory formalism and found that on the decoder layer of our network, if you seeded the T2 star ranges in a particular way, you were able to separate, for instance, the intra and extracellular water T2s and the free water T2s. So this is the right side is the successful NLS or SLED parameters, and the left side is if, for instance, the intra and extracellular water have very similar T2s as initialization of the parameters, not necessarily as their measured values, then you can't separate that. And that's borne out in the images here, which are actually from our data from a wild type animal. These aren't our best images, but they're for the purpose of showing that the information theory criterion for picking the initial values produces better MWF maps than if you use the conventional T2 star ranges for initialization, these are widely used in the literature in human 3T T2 star MWF, and I think it's a bit of a problem. So moving on to the actual SLED model, because it was a new deep learning method, we wanted to test it in an in silico phantom first. So Kevin came up with this experiment where he created a synthetic myelin water fraction phantom, where the vertical axis here is myelin water fraction from 0 to 0.5, and the horizontal axis is SNR up to 500. So you may not have 500 for your SNR in your images, but we're typically in sort of this range. And he could prove that that if you look over here at the error maps, you can see that the traditional NLS, even if you seed it with the optimal parameters from the ground truth map, it has a higher mean square error compared to the SLED maps. And this is a good property of the SLED because it tolerates low SNR data, and sometimes the later echoes in the multi-echo gradient echo data can have quite low SNR. I think I said mean square, it's mean absolute error here. So you see that there's a smoother transition for the SLED as a function of SNR for the mean absolute error. So this is the work that Kevin did in the actual mouse images from the Bruker system. They're a subset of the data that Vlad collected. This was a big effort, actually, that all the approximately 23 to 30 brains, some of them we didn't use, worked on by Marius Tuznik, who was an RA here, as well as Tim Wu. And I'll show pictures of them at the end, but they worked long hours to optimize the acquisition. And I won't belabor too much the mouse model, the MBPKO model. Vlad really elegantly explained this. The important thing to remember is that there's this graded hypomyelination phenotype with MBP mRNA values that decrease significantly in what's called the double knockout here, which we call 2KO, and the triple knockout. So there is this drop in MBP mRNA, which Vlad showed was correlated with myelin water fraction in different tracks going from the single to the double knockout. And this hypothesized increase in MBP mRNA in the knock-in, which we didn't see with the RPCA-based results. But since some people in the group do electron microscopy for verification of the myelin water fraction, I wanted to show these examples from Alan Peterson's group from the spinal cord. So this is our model. And what I like about this plot is it shows how the double knockout separates from the two M3 and M5 single knockouts as a function of axon diameter. So actually, in some respects, for larger axon diameters in the ventral medial spinal cord, based on these 1400 times TEM images, there's a larger separation here for the double knockout, and it's axon size dependent. So this is axon size along the x-axis and G ratio along the y-axis. And that's this is an example of the actual EM images from the double knockout, which Vlad alluded to, and he proved that you can see these G ratio changes non-invasively using MRI, but this is just sort of a gold standard comparison of the wild type and showing the thinning myelin for the double knockout in the lateral ventral cord. So Vlad mentioned, and I think it was Bruce or John asked about the bipolar readout specifically, and I'm going to describe our approach to dealing with this problem. As Vlad mentioned, we use a densely sampled bipolar multi-echo gradient echo readout, and the problem with that is that there's shifts in K-space for the even echoes relative to the odd echoes, and that can cause a mismatch in your data between your even and odd echoes. This is the same phenomenon that arises in EPI when you need to do row flipping. But in fact, what we needed to do for our data is we did row flipping, and then we implemented this method that was introduced for water fat separation in 2008 by Liu et al., whereby you're essentially trying to correct the shift along Kx. This is the K-space of a particular image, and the second echo has an offset relative to the odd echoes, and you try to correct this using a cross-correlation approach. Vlad's approach in his talk is slightly different from this, but it essentially tries to take this is the K-space coordinate or the K-space profile of the first echo, this is the second echo, and this is the third echo. So if you have 3 echoes, you can calculate this cross-correlation coefficient, and you can iterate on it according to your K-space shift in the readout direction to try to correct your data. And if you do this, this method is fairly robust to correcting the early echoes and the later echoes. Vlad alluded to the fact that you can either do this once for your first 3 echoes and apply that phase ramp to all your echoes, or you can do it independently for the later echoes. And the K-space for the later echoes looks a little bit different. So we applied it independently. And this is an example on the left of the uncorrected data showing the shift, the K-space phase ramp that Vlad alluded to. And on the right, after correction, the image does not shift as much. And you can see from the phase images of all 24 echoes that the phase accrues more linearly as a function of time if you perform this K-space based correction compared to if you just use the uncorrected Bruker phase data. So these are the parametric fitting results from SLED compared to NLLS, and generally the unsupervised deep learning model produced less noisy maps and showed more anatomical features compared to traditional parametric fitting methods with a 3-pool model. And these are examples of the graded hypomyelination phenotype, but now Vlad already showed this, but this is now based on the SLED model, the self-labeled SLED model. So both the SLED and the NLS-derived MWF match with the expected decrease in myelination across genotypes, but the deep learning model was less susceptible to noise, particularly in the gray matter. And this is an example of the actual tract-wise variations in MWF from the encoder versus NLS. What we found interestingly was that the traditional 3-pool model overestimated myelin water fraction, whereas SLED was closer to what we might believe to be the expected values. Kevin used slightly different anatomical parcellations from the Allen Brain Atlas than Vlad used. They're quite similar. He just did a few more. With the SLED model, interestingly, there was actually a slight difference between the hypermyelinated mouse and, for instance, the wild type and the M5KO, which we didn't see with RPCA. So we're still trying to tease out whether why we see this increased MWF value in SLED versus the RPCA approach. But by and large, across all of the phenotypes and across all the brain regions, we saw this decrease that we expect. And the decrease in myelination was different across different tracks. So when you're doing these kinds of new model fitting approaches, one sanity check that you can use is to noise your data using zero mean Gaussian noise. And we did this as well to try to determine the tolerance of the model for having noisy data. Maybe you scan for a shorter period of time, and the SLED mean absolute error was decreased significantly compared to the traditional methods. And this was also borne out in the numerical phantom. So this figure was produced based on synthetic data, again, the vertical axis being MWF across a large range, and now the SNR arrayed across perhaps a slightly more realistic SNR range if you're doing in vivo data, or if you can't scan for a very long period of time, like 6 hours overnight. And in this case, the encoder model improves in ground truth MWF estimation compared to NLS. So finally, I mentioned off the top that competing technique for fitting myelin water fraction mapping with these parameterized models is an artificial neural network. And an example of this was presented in really nice work by Zhang et al. And MRM in 2021. And of course, if you use an ANN to fit multi-component T2 star, you need training data. So that's the hang up there. And is your training data reliable, generalizable, or do you enter into a situation where you have overfitting? And this doesn't hamper SLED. But we implemented, I showed this figure previously, we implemented the artificial neural network approach just to compare sort of a pre-trained synthetic decay model to our SLED data. And what we found is that even if you justify the artificial neural network, that is, you pre-select your synthetic decay data based on the information theory resolution limits that Kevin came up with using same echoes, SLED generally outperformed both ANNs, the baseline and the justified. So to summarize, a variety of approaches exist for high-resolution multi-compartment T2 star MWF mapping. SLED is unsupervised and generally outperforms the existing methods. It relies on neural networks with automatic randomized initialization, and it was capable of identifying the graded hypomyelination that Vlad has nicely described already. So what we're planning to do in future is Kevin was interested in extending this to spin echo-based methods, multi-echo spin echo or GRACE, which may not have a purely exponential decay due to stimulated echoes. So he's seeking to develop a mathematically differentiable model of EPG for fitting MESE data or GRACE data. And as has been alluded to from a few questions in Vlad's talk, integration of the complex fitting model with local frequency shift values for intra and extracellular water pools, as well as orientation-dependent effects. So with that, I'll conclude. I really want to give all credit to Kevin here. He was my postdoc for a few years, and now he's got a faculty position in China. Vlad also did a lot of work on this and worked closely with Kevin. Tim, who's a research scientist with Camille Uludag now in Toronto, who did a little bit of the optimization on the sequence programming, as well as the Alan Peterson and Human, who are the brains behind the model, and Grace, who's an undergraduate who's working in my lab. And I also want to thank our funding sources, and I'm happy to take any questions.